Image and ORNLled team used neutrons to observe the AAT enzyme, a vitamin B6-dependent protein, and found that the chemical reaction occurred only in one active site. Nuclear scattering length density. View more. Credit Jill Heman and Andrei Kovalevska, Oak Ridge National Laboratory, U.S. Department of Energy, Oak Ridge. 10. October 16, 2017. Scientists at the Department of Energy's Oak Ridge National Laboratory have performed neutron structural analysis of a vitamin B6-dependent protein, potentially opening avenues for new antibiotics and drugs to battle diseases such as drug-resistant tuberculosis, malaria and diabetes. Specifically, the team used neutron crystallography to study the location of hydrogen atoms in aspartate aminotransferase, or AAT, an enzyme vital to the metabolism of certain amino acids. We visualized the first neutron structure of a vitamin B6 enzyme that belongs to a large protein family with hundreds of members that exist in nature, said ORNL's Andrei Kovalevska, a senior co-author of the study, which was published in Nature Communications. Vitamin B6-dependent proteins are part of a diverse group of enzymes that conduct over 100 different chemical reactions in cells. The enzymes are of interest to biomedical, as well as bioenergy, researchers because of their role in metabolizing amino acids and other cell nutrients. These enzymes are unique in that each one performs a specific chemical reaction with exquisite accuracy, while suppressing other viable chemical transformations, Kovalevska said. How they accomplish this is not well understood, but it is of great significance for drug design. The team's previous research predicted that hydrogen atoms move in and around the enzyme's active site, where the chemical reaction takes place, indicating that the hydrogen atom's positioning controls their reaction type. Knowing the precise location of hydrogen atoms can explain why the behavior of these enzymes is so specific, but hydrogen is hard to detect with standard methods such as X-ray crystallography. To directly determine the positions of hydrogen atoms within AAT, the ORNLled team turned to neutron diffraction techniques. The researchers exposed delicate protein crystals to neutrons using the Imagine beamline at ORNL's high-flux isotope reactor and the LADIII beamline at the Institut La Langevin in Grenoble, France. Surprisingly, the team observed the reaction within one AAT protein biomolecule while another AAT biomolecule was unchanged, providing a before and after perspective of the enzyme-catalyzed chemical reaction. The data revealed that in one of the enzyme's biomolecular structures the covalent bonds reorganized after a chemical reaction occurred in the active site and, in another, the reaction had not taken place, Kovalevska said. Essentially, we were able to obtain two structures in one crystal, which has never been done before for any protein using neutrons. With this knowledge, the team will run molecular simulations to determine the hydrogen atom's specific behavior when interacting with the enzyme. The results could be useful in guiding the future design of novel medicines against multi-drug resistant tuberculosis, malaria, diabetes and antibiotic-resistant bacteria. This study highlights how neutrons are an unrivaled probe for identifying the location of hydrogen atoms in biological systems, providing us with an unprecedented level of structural detail for this important enzyme, LADIII beamline scientist Matthew Blakely said. The neutron crystallography study, titled Direct Visualization of Critical Hydrogen Atoms in a Pyridoxal 5-Phosphate Enzyme, included co-authors Stephen Dajnovich of ORNL and the University of Toledo, ORNL's Andrei Kovalevska, Jerry Parks, Ryan C. Johnston and Kevin L. Weiss, Matthew P. Blakely of the Institute La Langevin, David A. Keene of Rutherford Appleton Laboratory, Oksana Gurlitz of the University of Tennessee, and Timothy C. Musser of the University of Toledo. Stephen Dajnovich, the paper's lead co-author and a doctoral student at the University of Toledo, works in ORNL's Biology and Soft Matter Division as part of ORNL's Graduate Opportunities Go student program. Musser is Dajnovich advisor and a senior co-author of the paper. This project was funded by DA's Office of Science and used resources at ORNL's Center for Structural Molecular Biology. Neutron crystallography work was performed at ORNL's HFIR, a DO Office of Science user facility, and at the Institut La Langevin. This work also used ORNL's Compute and Data Environment for Science for quantum mechanical calculations. This project was also supported in part by the Center for the Advancement of Science in Space and the National Aeronautics and Space Administration. ORNL is managed by Ut Patel for the Department of Energy's Office of Science, the single largest supporter of basic research in the physical sciences in the United States. DA's Office of Science is working to address some of the most pressing challenges of our time. For more information, please visit http://science.energy.gov. Disclaimer: AAAS and Eureka Alert are not responsible for the accuracy of news releases posted to Eureka Alert by contributing institutions or for the use of any information through the Eureka Alert system.